Hello and welcome to Sciencera and discuss peptide bonds. What is a peptide bond? A peptide is a short chain made up of amino acid which together with other peptides form a protein. Number of amino acids in a peptide can range from 2 amino acid up to 50 amino acids. Based on the number of amino acid present in peptides, peptides are of many types. Peptides with 10 or fewer amino acids are termed as oligopeptides, and peptides with more than 10 amino acids are termed as polypeptides. Polypeptides with around 100 amino acids are then considered proteins. Peptide bond definition. A peptide bond is a special type of amide bond formed between two molecules where an alpha carboxyl group of one molecule react with alpha amino group of another molecule, releasing a water molecule. The peptide bond is also referred to as a isopeptide bond, where the amide bond forms between carboxyl group of one amino acid and amino group of another amino acid at other position than the alpha. The process of formation of the peptide bond is an example of condensation reaction resulting in dehydration, which means removal of water. Peptide bonds are covalent bond that exist between any two amino acids resulting in a peptide chain. A partial double bond exists between carbon and nitrogen of a amide bond which stabilizes the peptide bond. Nitrogen involved in bond donates its lone pair to the carbonyl group, resulting in a resonance effect. This resonance is highly stabilizing as the electrons can be delocalized over multiple atoms, resulting in a resonance structure. Thus, the resonance structure stabilizes the bond but also limits the rotation around the amide bond due to the partial double bond. Peptide bonds have a planar configuration that undergoes very little movement around carbon-nitrogen bond but other single bonds on either side of carbon-nitrogen bond exhibit a high degree of rotational movement. Let's move on to the peptide formation mechanism. So, how is the peptides are formed? Mechanism of peptide bond formation is a dehydration synthesis process. During the formation of a peptide bond, the carboxyl group of one amino acid moves towards the amino group of another amino acid. Subsequently, one hydrogen and one oxygen atoms are lost from the carboxyl group of the first amino acid. In contrast, one hydrogen is lost from the amino group of the other amino acid. This results in release of water. Along with formation of an amide bond between two amino acids, the process of formation of a peptide bond between two amino acids results in a dipeptide bond. Thus, a peptide bond is formed when the carboxyl group of one amino acid condenses with amino group of another amino acid, releasing in a water molecule. The formation of a peptide bond is an endergonic reaction that requires energy, which is obtained from the ATP in living being. Because this reaction involves the removal of water molecule, it is called dehydration synthesis reaction. The degradation of the peptide bond takes place through hydrolysis, thus requires the presence of water molecule. Degradation reaction is very slow as amide bond with amino acid is stabilized by the partial double bond. Because of the partial double bond between carbon and nitrogen molecule, carbon atom generates a slight positive charge. In the presence of water, OH ions of water attack the carbon atom which result in degradation of the peptide bond. Remaining hydrogen ion of the water then attacks the nitrogen atom, resulting in the amino group. As a result of this, the peptide molecule is cleaved into two units, one unit with carboxyl group and another with the amino group. The degradation of the peptide is exergonic reaction that releases about 8 to 16 kilojoules of energy per mole. Because the protein degradation reaction are very slow, they are usually catalyzed by proteolytic enzyme like proteases and peptidases. Then we have peptide bond hydrolysis. Peptide bond hydrolysis is the primary step of all protein hydrolysis reaction. The most common method of protein degradation is acid catalyzed hydrolysis of peptide bond. The peptide hydrolysis is also essential in some synthetic reaction where amino acid in one peptide are cleaved and transferred to another peptide, resulting in separate peptide synthesis. Similarly, different peptides and protein accumulate in cell, resulting in toxicity. Peptide bond hydrolysis is essential in removal of those toxins as well. Peptide bond hydrolysis is also an important step in digestion of proteins in living being. Hydrolysis of peptide bond occurs in presence of water and is catalyzed by the presence of acid. Peptide bond hydrolysis is one of the mechanisms of peptide bond degradation where polypeptides are either cleaved into smaller peptides or smaller peptides are degraded into the separate amino acids. For example, the peptide bond is present in all proteins that binds the amino acid and chain together. So when you have one amino acid, it is monopeptide, two amino acids dipeptide, three amino acids tripeptide, six amino acids hexapeptide, and eight amino acids octapeptide, and so on. This brings us to the end of our peptide bond. Thank you for watching. If you like my video, don't forget to subscribe for more.